Hello everybody and welcome to my talk, which is entitled BioTink. So today I will tell you something about the iGEM project from Munich. But at first I would like to introduce the whole competition because I'm not sure whether everybody's familiar. So iGEM stands for International Genetically Engineered Machine Competition. So the idea is that you have teams of 5 to 20 students, undergraduate students, and they work the whole summer on a project, and then they all come together in Boston once a year, usually at the beginning of November, and then they present their scientific topics to a jury of judges, and then try to convince them that they did great science. And this kind of iGEM competition that started in 2003 is, in my opinion, a great opportunity for synthetic biology. It does not only fascinate the students, but it also offers the opportunity to really do prototyping in SynBio, which is often quite difficult because it requires lots of manpower, finances, lab space, and know-how. And this is usually not available if you come up with a great idea. But in this kind of situation with a team of 10 motivated students and the finances and lab space from the university, you can bring your idea from the first thoughts to a proof of principle in half a year. So we can expect there will be some nice outcomes from iGEM in the next years. So why am I standing here? Last year we were quite lucky in the competition. Usually there is a winner for the undergrad and for the overgrad section. And for the overgrad section, we were selected to be the winner, and the trophy is then such a Lego brick. And the other section was awarded to the Imperial team last year. And I've looked up, there was also a team 2 years before in Cork, so it tends to be in different universities all over the globe. But usually it starts with a certain problem, and the students of the team that I advised, they came up with organ shortage. So we could drastically improve the whole health situation if we would have enough donor organs. But as you see here from the numbers, these are cases or patients that are waiting for an organ to be transplanted, and this only in the US. So if we could provide these organs, we would improve all their lives. But sadly, the number of required organs far exceeds the number of donated organs. So the idea is quite simple. Why not to print them? And so the students had a look on conventional technologies, and most of the bioprinting approaches at the moment, they focus on a so-called hydrogel approach. So a macromolecule is used to form so-called hydrogel, a matrix, and in between these macromolecules, cells are printed. And after forming some extracellular matrix, at the end, these hydrogel can be dissolved and a kind of tissue is formed. The disadvantage of this process is that you need to dissolve the hydrogel at the end, and it's also quite laborious. So the students came up with another approach, and they thought, why can't we just make the cells stick to each other so that we can get rid of this collagen, this hydrogel? And so after discussing, we found biotin and avidin to be perfect molecules or binding partners to create such a two-component molecular glue. So the idea is that you have a bioprinter, and in your print head you have biotinylated cells, and then you print into a reservoir solution, and there you have a tetravalent binding protein, avidin. And as soon as the liquid from the printhead is printed into the reservoir solution, there's a formation of a cell network. So, and by this means, you can get rid of all this collagen and the extracellular matrix. And in theory, this should already lead to cells that stick to each other and can be printed. So let's have a closer look at these molecular components. So one of them is the avidin. You may know it as streptavidin, which is the bacterial variant, and it's a tetravalent binding protein for the vitamin biotin. And biotin can be attached to cells or proteins by different means. And the nice thing about it, it's one of the most affine tight binding known to biology that is not covalent. So what we needed for our approach was cells that carried biotins, and we needed avidin. The biotinylation of the cells can be done by chemical modification of the cells or in a SynBio approach using genetically encoded receptors or membrane proteins that code for the biotinylation. On the other hand, in theory, it's also possible to invert the whole system. So we could also put the avidin moieties on the cell surface and then link the cells using biotinylated proteins. So this is all we done. All we have done, and the beginning was chemical biotinylation. This is quite simple. This is one of the possibilities how it can be done. We have biotin NHS, and it simply binds to all amino groups of lysine residues. And if you take some GFP, which we had already in our lab, then you get a nice distribution of biotinylated proteins as shown in MS. And in average, we had 4 to 5 biotin molecules attached to each GFP. And this was then the protein that we used for bioprinting. But, and this can also be done with cells for sure, because the amino groups are also present on the cell surface. But on the other hand, this was a kind of genetic or SynBio competition, so we had to do it also by means of genetic engineering. And so what we are looking now at are these small membrane proteins carrying biotin moieties. And what we designed is a kind of membrane protein that allows us to present a biotin moiety that is attached to a receptor. And we did the whole design in a modular fashion so that each component can be exchanged, so that other researchers or iGEM teams can just create their own modular membrane proteins. In our case, we had a biotin acceptor peptide, which is a small peptide that gets biotinylated by an enzyme called BRA. It's a biotin ligase from E. coli. And this is restricted to the endoplasmatic reticulum of the cells. Then we had a folded domain, an epitope that was later on used for detection with antibodies, and we also added a fluorescent protein to the cytosolic part of the receptor. And what we see here is now the other receptor that we created for binding biotinylated proteins, and this one carries either a single moiety of avidin or a tetravalent single chain variant. And we tested all of these receptors in eukaryotic cells. We stably transformed them and afterwards did imaging. They all look the same. As an example, I just show you the single chain avidin. And as you can see, all the cells are transformed with our plasmid, with our genetic construct. In the DAPI channel, we see the nuclei stained with DAPI. Then we had some fluorophores attached to biotin, and they were nicely located to the outside or the exterior, the surface of the cells. And this was also true for the red fluorescent protein that was located to the cell surface, as well as the detection with a FAP that was fluorescently labeled that recognized an epitope tag of the receptor. We also did the quantification of these receptors in FACS, so with fluorescent activated cell sorting, and we could show that the red fluorescent protein was present at the cell surface. We also could detect the epitope tag, and for the receptors that had a biotin binding functionality, we also could show the binding of fluorophores that were connected to biotin moieties. So expression, localization, and topology were confirmed. Then we were also lucky that one professor offered us the opportunity to do a scanning electron microscopy experiment in which he correlated some fluorescent microscopic images, and we had some of our transgenic cells stained with magnetic beads that were conjugated with avidin, and what we see is that all these clumps, they accumulate in between these cells. So besides the biology, we also needed to have a bioprinter. And the idea was to have a bioprinter that is universal and that can be used by everybody. So our idea was to design printing files so that everybody could buy his own plastic bioprinter, download the files, print his own spare parts, buy some wires and some rods and some screws, and then assemble in less than 1 week and for less than $50 US his own bioprinter. So we hired a mechanical engineer, and he designed syringe pumps. He did stress models to see whether the stress that is applied to the plastic components is acceptable. He did nice documentation, and in the end he came up with a bioprinter. So you just can go into the internet and download the printer and get your own bioprinter that works with a small nozzle. And yeah, that's how it looked at the end, our bioprinter. So there was a dish retainer in which you could insert a small petri dish. We had a syringe pump which was able to deliver nanoliter volumes through the printhead that contained a small needle. And the only thing, if you have assembled your bioprinter to this stage, is that you upload a new firmware onto your bioprinter, and then you're done, and you can start bioprinting. So, so now we have the bioprinter as well as the components, and now we did some first experiments and looked whether this idea of a two-component glue really works. And the first experiment is just biotin-related cells, and they are dispensed into PBS. And what we see that there are some cells that clump, but in general, all the other, they don't really adhere to each other. But if we now add some tetravalent binding protein, we see that that there is nice stitching together of the cells, and they also stay together after being dispersed out of the needle. But what we learned here is that the whole process is really dependent on concentrations. So we also went to the mathematics department and hired a guy from there to do a mathematical model, and he came up with this model of ordinary differential equations. And what he found is that there is a sweet spot in which polymerization functions. And if we have too less or too much of our cross-linker protein, then we won't have any cross-link of the cells. So this was also confirmed in some experiments in which we just added the same cells together with different concentrations of the avidin, which is the cross-linker. And so the last thing that we did over the summer was then to really try to print some initial tissues or some logo. So we used our university logo and printed it with our printer. And what you see is here in the extracellular matrix, which is mainly biotin-related GFP that was also added to the protein, and some cells that were bright shining here. We didn't have enough of our transgenic cells at the end because this was done in the last week of this whole competition. But we see that there's a nice precision of the printing, and yeah, there was only a last question remaining. Is it also possible to print in different layers? Because we wanted to get to the 3D point. And so this was one of the last experiments that we did before heading to Boston. And so we used two different stained cells, one's blue stained cells and one's red stained cells. And if we move the focus now in the microscope, we see that only one of the cell types that were printed in one layer stay in the focus. So it seems that it's also possible to print different layers, but this would have been to be confirmed in confocal microscopy and with more than 2 layers. But yeah, that's where the experiment stopped in Boston. If we have a general look on advantages that this kind of approach could have over conventional bioprinting, we have lots of different parameters that we can adjust. So the affinity of the binding proteins, the kind of biotinylation, whether it's stable or liable, it's cleavable, concentrations of proteins, expression levels of the receptors, and also the cell densities. And I guess if one would optimize all these conditions, one could really come to nice results in bioprinting. At the moment, we are trying some business plan competitions and thought about a use case. And after looking at the market, we found that drug testing in pharmaceutical industry requires some small tissue explants that are then used, for example, for testing liver toxicity of pharmaceutical compounds. And yeah, we explored possibilities if this could also be done with our technology. Yeah, I have to thank lots of people, the team that did all the work, also some generous support from other professors, the professors at our universities, funding bodies at the university and in industry. And this was the team that did most of the work. And yeah, if there are some questions, I would like to answer them. Thank you for your attention.
So, I was wondering what you see as the next step. What's your role in this?
I guess, yeah, that's... Yeah, I mean, I will finish my PhD this summer, luckily, so yeah, that's a bit the problem about all these competitions. So you try to get everything done in half a year, you try to make it as complex as possible, and yeah, then you try perhaps to get it published, which will be done this year, I guess. But if you don't have a chair or a project or funding for this specific kind of project, it tends to become difficult to continue the work on it. But yeah, we are redoing all the cells, and we'll try to work 1 or 2 months to get it really published and to explore how good it really works, just for curiosity and for the publication. I mean, in iGEM there is this undergrad and overgrad, so if one of the students is over 23, then you need to go to overgrad. And if you don't want to exclude some students from the team, then in Europe usually you need to go for the overgrad section. So we were also in the overgrad section, Imperial was in undergrad, so yeah, it was approximately half undergrad, half overgrad. I mean, half from bachelor's, half from master's, and yeah, the advisors, yeah, it tends that people invest lots of time when they get infected once with this virus, and yeah, I mean, I also spent far too much time beside my PhD, but yeah, you learn a lot, and it's also nice for education, and if you like to work in teams, then this is perfect. But yeah.
Does the system work with different cells, or how do you pick which cells to use?
I mean, I just showed you the eukaryotic ones. So we had a recombination system from in vitrogen that worked with a recombinase, just because it was the most easiest way to get stable cell lines. In theory, with CRISPR, you could use any cell type. We used hex cells at the beginning, and what I didn't show to you was that we also did all these kind of binding molecules to prokaryotic cells, I mean E. coli. So there we used an autotransporter that could present all these functional modules at the surface of E. coli. But at the moment, it's only hex cells and E. coli.
Hi, Rory. Did you do any viability studies after? Because when you look at that kind of clump, and you think they would not be fairly viable.
I guess this will be one of the. I mean, we have bought all the reagents, and we had already the tests there, but due to time constraints, we couldn't grow the cells anymore. But usually what you expect also in this kind of hydrogel, the cells don't feel happy. And with all the experts that we talked, they were quite confident that it should be better than in a hydrogel. But this needs to be explored, yes.
Would you be able to show us where the schematics, etc., are from your 3D bioprinter? Are they available online, open source?
Yes, sure. This one?
Yeah. Is that on a particular website you can go to?
Yeah, it's on the wiki from iGEM. It's also on Hackaday, and there you can download the firmware for the Ultimaker 2 Plus, but also all the print files that you just have to incorporate into or load onto your bioprint on your plastic printer. So.
Thank you very much.